All right, good afternoon. I'm quite excited to share with you our work using nanoflares for the detection of circulating cancer stem cells. And my name is Chonghui Cheng. I'm an assistant professor in the division of Hem-Onc. And this is really a collaboration work among several groups, especially the lab of Dr. Stegh, who's really a driving force for this project. So breast cancer is the second leading cause of death in women in the United States. And each year there are about 40,000 women that die because of breast cancer. And it is known that primary tumor do not kill patients. Rather, it is tumor metastasis that still remain as the major cause of mortality in breast cancer patients, despite the many advanced therapeutics that are developed in recent years. So if we are looking at tumor metastasis, in fact, it's a multi-step process that is involved in many, many years. And you can see that first, tumor cells will grow as a nodule in epithelium, and then it is involved in the disruption of basal laminin, which I show you over here, and invade through blood vessels, through the extracellular matrix, going into blood vessel. And these cells that travel inside of blood vessel and go into distant regions. And these blood cells can then adhere to blood vessels, invade through the walls, and then colonize in distant organs. And these are metastatic tumors. And again, it is involved in multiple years for the development of metastatic tumors from the time that primary tumors can be detected. Now, recent studies have shown that in very early stage, there are these circulating cancer stem cells in the blood, and these are the cause of metastatic tumors. So it will be wonderful if we are able to detect these circulating cancer stem cells such that we can detect which are the group of patients that will eventually form metastatic lesions, and therefore we can establish better therapeutic strategies for those patients. Study of tumor metastasis has shown that those metastatic tumor cells have undergone epithelial-mesenchymal transition, also called EMT. And EMT is a developmental process that is involved in mesoderm formation, neural crest cell migration. However, this developmental process is also abnormally used in cancer cells for the migration as well as invasion into distant organs. What is EMT? EMT is involved in the change from epithelial cells that are cobblestone-like, that are non-motile, to mesenchymal cells that are spindle-like, very motile, and resistant to cell death. It is known that there are several drivers for EMT. One of such is called TWIST, which I show you over here. It's a transcription factor. If you express TWIST in epithelial cells, those cells will undergo EMT to mesenchymal state. And it has also been shown that the TWIST can drive tumor metastasis in animals and highly expressed in metastatic tumors. Studies of EMT have also shown that there are specific markers for epithelial cells as well as mesenchymal cells. For example, E-cadherin is expressed highly in epithelial cells and is not expressed in mesenchymal cells. Conversely, vimentin and fibronectin are markers of the mesenchymal cells and is not expressed in epithelial cells. So what's interesting is that cancer stem cells have been shown to show properties of EMT. Now, that gets together, what it means is that circulating cancer stem cells show property of EMT, and that is important for tumor metastasis in patients. That leads us to ask the question, can we detect cancer stem cells in the bloodstream using EMT as a readout? So at this moment, the cancer stem cell detection is quite limited because of the low amount of circulating cancer stem cells that one can isolate. For example, people can use antibodies to detect cancer stem cells. However, that relies on the expression of certain proteins on the cell surface. So to get around with this, we decided to ask whether we have any ways to detect mRNAs using technologies in live cells. And that comes with the technology of nanoflares, which enable us to detect cancer stem cells using mRNA methods. Showing here is a nanoflare particle. You can see the gold particle that's conjugated with DNA oligos. So this is a strand, the green is a strand that's a recognition strand for a sequence that you're interested in, for example, E-cadherin, if I give a name of an mRNA. And the blue is conjugated with the color red showing over here. So red is quenched because of the close distance with the gold particle. And I will show you how this method is working step by step. So again, there's this gold particle conjugated with a DNA of recognition sequence. For example, let's say E-cadherin. And then we have a reporter flare sequence that's linked to a color, for example, Psi5, that shines red. Once these two are combined together, because of the duplex formation, because this is a complementary to the recognition sequence, this duplex will quench the red color due to the distance between the Psi5 and the gold particle. When we introduce this nanoflare into cells, the target mRNA, such as E-cadherin, which is the same DNA sequence with the reporter flare sequence, that will kick off the reporter sequence, making it far away from this nanoparticle such that it starts to shine red color. And what we can do is we can quantitate the amount of red color for the measurement of how much E-cadherin is expressed inside of cells. So again, showing here is a nanoparticle with the DNA oligo conjugated as a nanoflare. And this technique has been developed in Dr. Merkin's lab and has been used for the detection of surviving mRNAs in cells, which I show you as an example. So this is the picture using survivin flare and looking at the expression of survivin in cells, and you can see red pretty nicely. If you are using a non-complementary flare as a control, you see that essentially very low levels of red is detected. And showing here is a quantitation of the fluorescence increase comparing the cells using surviving flare mRNA or the control nanoparticle or control non-specific mRNAs. Now, with all this in mind, we decided first to use a model system of EMT, first to develop nanoflares that we can look at the markers of EMT. And what we did was to use a human memory epithelial cell line called HMLE, which is epithelial looking, as well as a mesenchymal line that is derived from HMLE by expression the transcription factor twist. As you can see that these cells have undergone EMT with a spindle-like structure. And the mRNA targets that we have been using are E-cadherin, which is highly expressed in epithelial cells, and twist, which is highly expressed in HMLE twist cells. And these are the two nanoflare particles that we have designed. One is to using the E-cadherin flare conjugated with a Psi5 color, and the other is a twist flare conjugated with a Psi5 color. We then introduced these flares into two cell lines. One is a HMLE, and the other is HMLE twist, serving as epithelial and mesenchymal cell models. And looking at the HMLE epithelial cells, supposedly E-cadherin level should be high as it's an epithelial marker. We were able to observe about threefold upregulation of E-cadherin signal. However, we really did not see much of the enrichment of the signal from twist flare, suggesting that these cells express E-cadherin rather than twist. By contrast, if we are looking at the HMLE twist cells, the twist flare shows about 15-fold enrichment of the fluorescence as compared to control. And we essentially do not see any of the signal that is enriched by using E-cadherin flare. So these results suggest to us that the E-cadherin flare and twist flare both work in the epithelial and mesenchymal cells, and they can be used as a readout for detection of the expression of E-cadherin and twist in these cell lines. And these are our model systems. So we decided to move further, and we wanted to use more markers for the detection of metastatic tumor cell lines. And here we used MBMD231 as a model system, which has been shown as a breast cancer metastatic cell line. First, we developed 2 pairs of vimentin and fibronectin flares. And these are genes that are highly expressed in mesenchymal cells. And what we did first was to validate whether the flares work in tubes. We incubated the mRNA of vimentin or fibronectin together with a NanoFlare. And we are able to see that they are quite specific and efficient. So for both of the vimentin flares, there is an enrichment of more than 15-fold. And for the fibronectin, you can see about a 10-fold enrichment for the color as compared to the control. So that says to us that these 2 flares, 2 sets of flares work. Then we tested it in cell lines using the 231 line and incubated with NanoFlare, either fibronectin or vimentin. As you can see from this plot, the fibronectin flare shows about 6-fold enrichment as compared to control flare, and the vimentin flare showed about 9-fold enrichment in the color as compared to control flare, suggesting that these flares do work to detect the levels of vimentin and fibronectin in the cell lines in tissue culture. Now, we then decided to ask, can we combine with human blood to get it to the real world and to see whether we can detect a very limited amount of tumor cells in the whole blood? The scheme is the following. We have volunteer blood, so these are healthy people, and incubated with known amount of M-cherry, which shines a red color of the MDMB231 cells, and then also incubated with a NanoFlare, for example, targeting vimentin or fibronectin. And then we went through the whole process by incubating 8 hours at 37 degrees, going through CD45 depletion to get rid, because in the whole blood, the major things are the leukocytes. Then we want to get rid of those. We wanted to enrich for cancer stem cells. So we did the CD45 depletion and the FICO gradient, as well as later the red blood cell lysate. And then we went on flow cytometry and to see the color, because the flare has its color and M-cherry also has its color. What we can see over here is that in both, let's just look at the left side using the vimentin NanoFlare. We can see 2 peaks. One peak, these are the cells that essentially show very low levels of NanoFlare Cy5 color. And this is another peak that shows extremely high levels of fluorescence in a NanoFlare color. And similar observation is shown in fibronectin NanoFlares. What's interesting is that the MDMB231 cells are essentially located all in this population. And these are the leukocytes, all those blood cells that are shown over here. But about 99.3% of the recovered 231 cells are independently independently detected by NanoFlare. Again, suggesting that this is really a robust method to isolate cancer stem cells. And this is just a plot for the fluorescence enrichment using the vimentin and the fibronectin flares. And you can see it's highly enriched in the MDA and MB231 cells. So the next question we asked is, what is the limitation for our detection? Can we really detect extremely low numbers of circulating cancer stem cells? To do that, we did a serial titration of known amounts of MD and MB231 cells. Shown here in orange are the uncherished cells. And the APC, this axis, shows the flare signal. And what you can see is that using 10,000 cells, 1,000 cells, and even 100 cells, you are able to detect the uncherished labeled 231 cells in the whole blood, in a healthy patient whole blood samples. We plotted the cell recovery rate, and what we saw was that essentially this is the number of cells added, and this is the cell number that's recovered. What we can see is that about 70% of the cells can be recovered using this method. So again, what we found is that this is quite a robust method to isolate the cancer stem cells in the circulation. So the next question that we asked is that, well, the limitation is really low, so it's really sensitive, and whether through all these processes, which is involved in 8 hours and longer, do these cancer stem cells still survive? Can we really test whether these cells can be used, for example, for the future therapeutic development or research studies? To answer this question, what we decided to do was to go through the entire process and then assay for cancer stem cell properties. And over here, we decided to do memory sphere formation, which is one of the readouts for cancer stem cells. So again, we have volunteer blood samples and incubated with known GFP recurrent tumor cells, and with the NanoFlare. Over here, we used the vimentin. And then we incubated again 8 hours, going through all the blood cell depletion and went on the FACS. And then we retrieved the cells and put them into the assay for the memory sphere formation. So showing on the left are the FACS analysis, where again, we can see that there's an enrichment for vimentin high nanoflares in the recurrent cells that shine green. And then we retrieved all these cells and put them into the dish for memory sphere formation. Much to our delight, what we can see is that for the retrieved cells, they are still alive, and they have the property of forming forming the spheres in suspension, which you can see over here. So these are the spheres, and they do show green because these cells are GFP labeled, suggesting that for the limited amount of circulating cancer stem cells that we can isolate, they maintain alive even after more than 8 hours of processing. So with all this data in hand, we were encouraged to move forward to use breast cancer patient samples and to test our technology. Here, with the help of clinicians, especially Teri Odom at the Hem-Onc division, we were able to get several patient blood samples, and then we incubate these samples with nanoflares, and then went through all the process and detected the levels of nanoflares. So showing here are about 6 or 8 patient samples what you can see is that this is stage 0, so normal, and stage 2, 3, and several of the stage 4s. With the increase of the stage, you can see the increase of the nanoflare intensity, suggesting that the more the advanced the patient is, the more flare signal that we are able to detect from their circulating cancer stem, from their circulating blood. So these are the blood samples from patients. And the second thing I wanted to point out is over here. So this is a patient that we did 2 rounds of detection. The first time, we saw about 482 cells per ml of this patient. And the second time, we saw about 300 cells per ml in this patient. So it's in a ballpark, pretty much consistent. The second thing I wanted to mention is that this patient is undergoing chemotherapy. So we were wondering if this slight drop of the cancer stem cells in the circulation correlates with the therapy that she's taking. So we are interested in following this patient up and see with time, would we be able to see the drop of the flare signal with the treatment that she's receiving. So it's quite encouraging at this moment. And we are collecting more patient samples and trying to see whether this is really a robust technology that we can really push into clinical diagnosis as well as future treatment. So in conclusion, we have optimized the nanoflares using our cell culture system. And we have also using different cancer cell models for the detection of nanoflares in metastatic tumor cell lines, as well as in an animal model, which I don't have time to show. And further, we have used this nanoflare technology and validated a few patient samples which have a metastatic potential. So at this moment, we are continuing working in this direction and hoping that one day we will be able to develop this technology and bring a new way of detecting circulating cancer stem cells in patients. So with this, I'd like to acknowledge the big teams. It's a few labs collaboration, especially as I mentioned to you earlier, the driving force is Dr. Stegh, which really this should be presented by him. And many of the postdocs and graduate students contributed to this work, which I listed over here. Kaylin Nick from Shad's lab, Tiffany and Alisa from the Merkin lab, Wei Wang and Yilin Xu from my lab, and Dr. Jin is the clinician who really gave us a head up for the clinical trials using patient samples with this nanotechnology. And special thanks to the CCNE program for funding. Thank you very much. I'd like to take any questions. Yeah.
Beautiful work.
Thank you.
I'm just wondering, the vimentin as your target is very much like a digital, it's on or off. Do you think the nanoflare technology can also be applied to systems where you're looking for quantitative differences in the expression between cells of other markers?
Yeah, so Paul's question is essentially how sensitive it is. Can we do the same like RT-PCR? For example, we can know exactly what amount of the mRNAs that we can detect. I would say at this moment, we are using the all and none mRNAs for the detection, and with the hope that in the future, we will be able to quantitate them just like RT-PCRs. Yeah. All right, I guess I will hand to Alex for the next talk. Thank you.